Yes, great. It's my pleasure to introduce the first speaker, is Amandine, who is a postdoc in my lab in EEB. Amandine's been doing a ton of really interesting work thinking about how to connect virology to transmission biology and fitness of some of the interesting viruses we've been wrestling with over the last year. And today she's going to show some great new work on how to think about dose response modeling in animal exposure experiments. Go for it.
Thanks, Jamie. And yeah, thanks QCBio team for the opportunity to present some of my work to the community. So yeah, as Jamie said, I'll be talking about linking exposure dose to infectious disease dynamics using mathematical models. So really linking aspects of virology and epidemiology and transmission dynamics in between. So those response models are not specific to infectious disease biology, and they're used to address a really wide range of, they're used in a wide range of fields to address questions of how much an event will occur depending on the dose of an exposure factor. So for instance, it's widely used in pharmacology to test the effect of drug dose on the response you expect in the host and on the pathogen or the disease. You can also find it in immunological studies, and I will more specifically focus on the relationship linking pathogen dose to host response in the context of infectious disease. So describing the dose-response relationship in a specific host pathogen model can have several objectives depending on which system you're considering. But when we're working on laboratory animals, one of the most common use of this kind of model is actually to optimize laboratory experiments. So for instance, if you're trying to test a vaccine, you need to identify which dose you're gonna inoculate, which dose of virus or other pathogen you're gonna inoculate to your animals to make sure that the ones that are not vaccinated will actually show the effect you expect from infection. So it can be either transmission of the virus, illness, or up to death of the animal, and with the hope to see this effect reducing in the presence of a vaccine or another treatment. It can also help you to define how many animals you want to include in your experiments depending on the variability. So do all the animals exposed to the same dose show the same response, or is there any individual variability within your system? And also how long to conduct the experiments if the incubation time is governed by the exposure or inoculum dose. So I talk about incubation time just to make clear, we all have the same definition in mind because we heard lots of those words recently, but people are sometimes using them with different definitions. So by incubation, I mean the period between the time you were exposed to the infectious agent or you inoculated your infectious agent to the animals, and the time where symptoms will appear. So it's the asymptomatic phase at the beginning of infection. And if you have an incubation, it means at some point you have a transition to a symptomatic phase of your of the disease dynamic, which correspond to illness onset. It's also describing the dose-response relationship in one system is also then the basis to test lots of effects that can have really important biological implications. So you can compare the infectivity of different variants, for instance, if we're thinking about the emerging variants for SARS-CoV-2. Is this dose-response relationship switched to a higher infectivity of the variant or lower infectivity? You can test effect of risk factors on host susceptibility, so either age or sex or comorbidities. You can test the effect of vaccine and antivirals, and you can also compare the susceptibility of different host species, and it can be really important when you're thinking about zoonotic diseases where you have several species that can be involved in the transmission chain of the pathogen. Once you have established a dose-response relationship in one system, you can also use it to get some information in other systems. And that's obviously really interesting when you're thinking about diseases that are harmful to humans and for which we want to have information, but we cannot obviously test it directly in humans. So from the information we can get in laboratory models, we can refine our epidemiological models by linking shedding to infection, because that's really what's happening when you consider the dose-response relationship. It's the missing step between what will explain that an individual get infected from what it was exposed to from the person that transmitted the virus in the first place. And also it goes into this framework of using more quantitative approaches. So typically in epidemiology, you know, individuals are classified in compartments of susceptible, infected, and recovering individuals. But if you consider the dose-response relationship, you could imagine having people who are shedding different quantities of virus, which will lead to different transmission probabilities for these individuals and so on. And also from a practical point of view, lots of the non-pharmaceutical interventions that we're using right now in the context of SARS-CoV-2 are actually aiming at reducing the inoculation dose. So that's really based on like implicitly based on the fact that we have this relationship between inoculation dose and probability of getting sick or getting infected or having a severe disease. So for instance, if you think about protective equipment that you either put on the source when people say you have to wear a mask to protect others, or that you put on the recipient when people say wearing a mask also protects you, it's actually so when you're infected, you shed less virus, so people are receiving less virus. And when you're wearing a mask, you're receiving even less. And it's the same when you think about the contamination of the environment or using ventilation to reduce the viral load in aerosols, or even just physical distancing, because you reduce opportunities for accumulation of virus transmission. Regarding the data, so far I've been talking about concepts, but there are data in the epidemiological world that are suggesting that there is a dose dependence of illness probability and incubation time when thinking about SARS-CoV-2 in humans. So you can see, for instance, on this plot that was published a few weeks ago from a really detailed cohort study in Spain, that the higher the dose the people received, and the more likely they were to have symptoms from SARS-CoV-2 infection. And also the higher the dose and the shorter the incubation period. So majority of the people who received the high dose had symptoms by 10 days, while it was a bit longer, up to 20 days in that case for the lower inoculation dose. And I'm talking about SARS-CoV-2 here because that's the system I've been working on, and that's the thing we hear a lot at the moment. But lots of the concepts are actually true for lots of other pathogens, if not all, and notably Salmonella, viruses causing gastroenteritis, or anthrax, plague, tuberculosis, and so on.
Amandine, can I just jump in with a question?
Yeah, no worries.
So you've been focused, and you're in your presentation, you focused on incubation time.
Yeah.
But there's also this issue of whether you might be infected but never become symptomatic.
Yeah. So it's actually a stepwise thing. So
right.
It's first you get infected, and depending on what, like if you're infected, depending on whether you're infected, then you will have a shorter or longer incubation time.
Yeah.
Yeah, I will detail
the data also shows, the fraction of symptom-free.
The fraction of what?
Of being of remaining asymptomatic, right?
Yeah. Some people will get infected and never show any symptom, and some people will not even get infected. So there's like a whole range of outcomes from infection, depending on the dose, but not only on the dose, obviously.
Right. So in your analysis, you will consider that.
Yeah, I will detail that in a few slides. So actually related to what you were just saying, the classical dose response models that are used in epidemiology are usually focused on that whether you get infected or not, whether you get symptoms or not, and whether you die or not, but not on on the time or other variable, really just on the binary outcome of exposure. So we usually obtain this kind of plots where we have the proportion of individual responding, so showing symptoms, transmission, and so on, plotted over the dose of pathogen they received on a log scale. So that's already bringing lots of information, but it's not considering all the heterogeneity there is in inoculation outcomes, and potentially the one that can be driven by inoculation dose. So as described before, when you get infected or when you are inoculated a pathogen and you get infected, you can incubate for a few days and then become symptomatic and eventually die in some cases. But some people will be infected, never show any symptoms. Some people will get infected, show symptoms and recover. And if we consider all this heterogeneity in the inoculation outcomes, that can help us first understanding where this heterogeneity is coming from. Notably, is it coming from the dose they received? It can help us also methodologically speaking, because we have then wider diversity of outcomes to consider in our analysis, give us more power to detect some effects. And another thing is timing is important. So that's why I've been focused on incubation time, because for the host, the longer the incubation time and potentially the more time the host will have to mount an immune response and fight the disease, which can, for instance, lead to less severe disease. And also it regarding applications in today's world, it's critical to understand how much can incubation time vary to design efficient quarantine protocols. And again, the more heterogeneity in the data and the more power we have to detect effects. So the system we're considering, I've been working on SARS-CoV-2, as you understood now, and on laboratory model, and especially on mouse model expressing the human receptor for the virus, which is a commonly used model for SARS-CoV-2 now. So we start with a dose. So we are in an experimental framework. So we start with a dose that we know. And the mice will be inoculated with this dose of variants. And some of the variants will successfully lead to an infection, while some will potentially never really enter the host or just get lost in the tissue and never encounter a cell and generate an infection, or they will be attacked by immunity before they get a chance to infect a cell. So we have, from the dose we started with, we only have a proportion of that we call successful variants. And then these successful variants, they can initiate viral replication, which will lead to the growth of a viral population, where we have entry with replication, and also some variants will be like a degradation rate coming from immunity and natural degradation of the variants. And at the outcome of this is what we observe in the host. So the effect here, we are thinking mostly about the symptoms. And so the parameters that will link all this together, as I mentioned, are the probability of successful infection for a variant that will link the dose to the founding population of variants. The viral population dynamics that will link how you get to a grown viral population when you started only with a few variants. And finally, the tolerance to the disease. So the propensity for individuals to declare symptoms based on the viral load they have in their body. So the hypotheses we are considering in this system are first that the probability of infection and illness are dose dependent, and notably the higher the dose, the higher those probabilities. But also that the incubation period will be dependent on exposure dose, and the higher the dose, the shorter the incubation period. We also expect that the dose-response relationship will be impacted by some host factors, in particular susceptibility and tolerance. So how much they're going to get infected by the virus and whether they're gonna declare symptoms or not. And so we need, one example, typically from the epidemiological data, we know that males are more likely to have severe disease. So is it because they're more susceptible and or less tolerant than females to SARS-CoV-2? And so to link all this together, we need a model that accounts for both the infection and the illness process and time, like on a temporal scale. Before moving to the data, I want to highlight that the data I'm using for this project are coming from collaborators at Cornell who are doing lots of work on SARS-CoV-2 and have been working really hard in the past week. So I'm really thankful that they're getting those data and sharing them with us. And so in particular, we're working with Hector Aguilar lab in the Department of Microbiology and Immunology. So another point I want to raise about this data is that they are opportunistic data that they originally collected as part of the optimization of a vaccine trial. So they were not initially collected with the objective of building a model on dose response relationship. But they remain experimental data, so they have lots of value like experimental data would have. So as I said, they infected HAS2 mice with SARS-CoV-2. So they used an old variant, so the one we had originally, like earlier in the pandemic, and classically use HAS2 mouse model. They considered 5 doses, so 10 to the 1 to 10 to the 5 plague-forming units. So that corresponds to it's a proxy of the number of infectious variants you have in the sample. And they also have a group of not infected individuals that I call mock. For each dose, they consider both females and males. And we had overall lots of individuals, but per group, only a few individuals with 3 to 5 individuals per sex and dose. The data that they collected are first the daily weight and survival. So each day they would check on the mice and weight them. I want to highlight that those data are non-invasive. So from an ethical perspective, it's even if it might seem unreasonable to try to fit a mechanistic model only on symptoms data and not using viral load data that we could get from swabs, for instance. Those data are non-invasive, and so from an ethical perspective, that's really interesting, and we'll see that it's still containing lots of information. So individuals were classified as symptomatic or asymptomatic, depending on their state when they were observed. And we don't have those data yet, but they also collected at the end of the experiment. So either when the animals got too sick and had to be euthanized, or at the end of the experiments where they were euthanized, some tissue samples that would be tested by PCR and histology. And this will be interesting to answer your question, Alex, to detect the individuals that were infected but didn't show any symptoms. So the one that we might miss if we're only focusing on the symptoms. And so here are the data that we got from them. So you can see the mock individuals are maintaining their weight all along the experiments. You can see that the males in blue are heavier than the females. And you can see that as the dose increase and as the day pass by, some individuals will start losing weight, both in males and females, and up to, if you're considering the higher doses, all individuals at some point starting losing weight, so they got really sick from the infection. If we look at it might be easier to look at the weight in terms of relative to the initial weight of the individuals. And so you can see that the weight loss can reach up to 20%. So that's actually the point at which they had to euthanize the animals, because it's considered not ethical anymore to keep individuals that are that sick from a disease. So that's why all the points are stopping at after this weight loss. And sometimes they also had additional symptoms that required euthanasia before they actually reached that endpoint of 20% weight loss. So just looking at this, we can already see lots of information. So the first one is that the probability of getting symptoms seems to increase with the dose. So for the low doses, we had 10 to the 1 or 10 PFU. We had no female getting sick and only 2 over 5 males. And this increased, but we quickly reached the doses at which all individuals got sick. We can see here that we have a recovery too. But the other interesting pattern is that if you look precisely at when the animals got sick, you can really see a really clear pattern where there is this asymptomatic phase that lasts for a few days, and then a quite brutal transition to a symptomatic phase where the individuals start losing weight. So that corresponds to the incubation period. And you can see that this incubation period is actually decreasing as the dose increase. So animals that received a higher dose got sick faster than the one that received a lower dose. So now I'm going to move to the mechanistic models we are building based on those data. And I want to first disclaim that I'm new to some of the aspects of this modeling exercise, notably all the Bayesian statistic aspects. So I received lots of encouragement and help from my colleagues in the Lloyd-Smith lab, and in particular, Dylan Morris and Benny Bormans, who are 2 other postdocs in the lab. So if we go back to our infection process that I described at the beginning, we can put variables now on those words that I mentioned. So we start with the inoculum dose that we consider we know. Then we have, after different steps of the infection process, we reach a founding population that we call V0 for viral load at time 0. Then we have the viral load that will increase with time based on the population dynamic of the virus. And we have the effect of the disease. So what's linking the initial dose to the founding population? We can imagine the Poisson process where each so we expect to have this V0 number to be a direct consequence of the initial dose, but with some loss that happens following some kind of random trials or like a binomial kind of situation where each variant has either a chance to get lost or to actually infect a cell. So that's the probability of actually infecting a cell. That's what I call P success. Then when starting from this founding population, V0, you will have, we can assume at the beginning of the infection process, exponential growth of the viral population. So we can model that pretty easily with like that simple equation, where the only additional parameter is that K parameter, which will represent the viral population growth rate. And finally, regarding the effect, so here symptoms or no symptoms, we can classify individuals as either they were sick or they were not sick. And the link between the viral population and the fact of showing symptoms is actually where we need to think a bit harder. So we'll focus on what's happening here. So each individual is either asymptomatic or symptomatic, and it will transition from asymptomatic to symptomatic after some time, potentially or not. And this transition rate will be given by a function of time that is actually itself a function of the viral population. So we considered that the higher the viral load within the host, so following replication and so on, the more likely an individual is to show symptom. So we have this time-dependent transition rate from asymptomatic to symptomatic. To start with, I've considered a simple linear relationship, but we can consider more complex relationships too. So this mechanism that I described, it's a typical continuous time Markov chain, where we have a variable rate, because it's this rate of transition is varying with time, Poisson process. And so we have the disease, non-diseased, or symptomatic, asymptomatic, or individual states, and transition events corresponding to becoming symptomatic, and incubation time can see at the waiting time. And then at each time where you observe an individual, if you observe that it was sick, it's because this rate was, you have to consider all the opportunities individual had to get sick between the infection and the observation time. So we will actually integrate lambda over our function here over time. And at the end, we can quite simply calculate the probability for an individual of not being symptomatic as the probability of not having transition yet, which is given by this exponential based on the Poisson process, or the probability of being symptomatic is just the complementary probability of this same process. So we actually have, even if we consider some relatively complex mechanism, we end up with relatively simple equations. So what I did so far is I simulated some data to check that the model we developed was making sense. And I will now in the next steps that I'm currently working on, use this simulated data to test the fitting performance of the Bayesian model I'm developing and that I will describe right now. So that's what you cannot observe, but what's happening in our host, in our simulated host. So we have the growth of the viral population, which leads to an increase in the probability of showing disease and individual transitioning from not disease to disease, with all this being governed by the initial dose. I do all the simulation in R. And another interesting thing to consider in this model is the observation process, because we don't see the actual time where an individual starts showing symptoms. Well, we see that one day wasn't sick and the following day was sick. So I won't go into the details, but based on the probability that we calculated before, we can quickly estimate the probability of observing an individual sick for the first time a given day based on a latent parameter that we don't estimate, but is happening in the model. That is the true incubation time. So I chose to use a Bayesian approach because it gives lots of flexibility. It allows you to propagate the uncertainty at the different stages of the process. And you can also use prior information, if you have some, to improve model fitting and optimizing your use of the data based on previously available knowledge. So I won't go into all the detail of the Bayesian model, but briefly we have this data set that consider everything from the observation to the experimental conditions. And we have the 3 parameters that I presented before. And the potential values for the parameters are linked to the observed value for the data based on the Bayes theorem. And this bold part is actually all the equations I described in the previous slide. So we have the information we need. I do model fitting with R and stat. And so actually I'm at the learning stage. So I'm working on the models. I've started fitting some really simple models and I'm moving to the more complicated one currently. So I don't have any fancy convergence of chains to show or stuff like that. But I hope you were interested in the framework. And so, yeah, for the next steps, we'll consider potential effect of sex and individual variation on the different parameters. Also test some biological relations, so notably whether we should consider the absolute number of variants when we consider the probability of getting sick, or should we consider the number of variants corrected by weight, which would first help us potentially improve model fitting, but also bring us some insights of what is really important in the biology of this dose-response relationship. We can also use directly the weight data instead of using a classification that requires to have a threshold between what is symptomatic and asymptomatic individuals. And we can try different functional shapes for the different equations. We could also, on a longer term, think about including disease severity and potentially see whether disease severity and weight loss over time can directly be used to parameter the viral load within the host. So in the context of the SARS-CoV-2 pandemic specifically, there are some longer term questions that we could think about. I think one of the most pressing one at the moment is how the different variants are impacting the dose relationship, because it seems like some of the emerging variants have a higher transmissibility, and it's important to know if it's due to more shedding or higher infectivity. We are thinking right now it's more higher infectivity because we have increased receptor binding within the host, but it would be interesting to have quantitative and experimental data on that question. And if we keep on even thinking further, I think there are lots of applications, not necessarily in the specific field of dose response modeling, but trying to link microbiology and epidemiology using quantitative models, notably because both are fields where it's mostly the transition between those fields tend to be reduced to, sick nor sick, or transmitted, didn't transmit. So all the data are resumed or reduced to binomial data. And if we had a more quantitative approach, we could have a better understanding of the mechanism at play, and also improve parameter estimation and statistical power. And so one of the practical objective of this kind of model that I'm developing is also to make some tools available for the microbiologists we're working with, like codes, packages, and applications. And overall, I think it's one step towards the big and ambitious objective of having a more quantitative and multi-scale understanding of epidemiological dynamics from what's happening regarding receptor binding up to the dose response relationship, and ultimately the transmission dynamics between humans and between animals and so on. And to conclude, I want to again acknowledge my colleagues at the Lloyd-Smith lab here at UCLA and also at Cornell, who are producing the data and helping me think about this model. And also really warmly thank Rana Plowright and all the people involved in the Bat One Health project for actually putting us in touch with the virologists. So we have nice data to work with and interesting questions to address. And I thank you all for your attention. And if you have any question after the talk, you can reach me by email or Twitter. Thank you.